Hi everyone, welcome to week 2 for microbiology. So this week we have 3 different PowerPoints that we're going to go through. We're going to really focus on some staining processes, especially gram stain. Again, it's our primary stain we use in micro, and then a little bit on a couple others. And then we'll really jump into learning some of our augers that you're going to get to play with in lab. So we'll first start with the microscopic examination PowerPoint. So again, there's objectives here for the PowerPoint itself on like what you should kind of get out of this PowerPoint. You can read those as you want. So different types of microscopy in general, just across all department areas, but you know, bright field is our main microscope we use in microbiology and hematology. There is also fluorescence, phase contrast, dark field, and electron. So I'll kind of mention a few little things on the rest of those. But bright field is the microscope that you're using, the style microscope you're using in your lab room, as well as in the micro department and in the heme department. So what it means to be a bright field microscope, it just means that there's light passing through the specimen and then up through the lenses. So when we think of our magnification, we have total magnification, which is your objective lens power, which is like around say 10x times your ocular lens power. Which the ocular is the one is your eyepiece, if you think of it that way, ocular for eyepiece. So those lenses are the ones that are around 10x. Objective can either be 10x, 40x, 100x. There's sometimes 50x on microscopes, especially in like hematology. In microbiology area, anytime you're using something that's stained, we're typically using 100x for the stained. 40x would be looking at something like a urine that's not stained. So there is a little difference there. But again, to come up with the total magnification, it's your objective times your ocular power, and that's the total magnification. Resolution is a term that just means clarity, how much you can see details on that, whatever you're looking at under the microscope. How clear is it? How fine-tuned are those details that you're able to see? That is the resolution. So those are both terms that you should definitely know and take away. Again, anytime we're dealing with stain stuff like gram staining, we're looking at the oil 100x objective. So in order to use the 100x oil, we do have to use immersion oil. If you've already taken hematology, you should be familiar with this. We did that as well in hematology, looking at differentials. So that immersion oil is just used to fill up the space between the 100x objective and the slide. And it just enhances that resolution, that clarity, ability to really visually see things well. So again, we are using bright field microscope to identify bacteria, fungi, parasites, again, typically stained items for sure. So staining methods, before we ever start any kind of staining process, you have to first fix whatever you're looking at to the slide. So you use a clean slide, and typically what you're going to learn probably in our micro lab time is going to be like a drop of saline, a drop of DI water, something like that, but very small drop. The smaller the drop, typically the better. Otherwise, it takes a long time for it to dry. And then you mix in, like, say, your bacteria into that, and then you're going to let that dry. Once that is air dried, you need to fix it. And you either fix it by heat or you'll fix it by a chemical like xylene. And the fixing it makes sure that when you go to stain it, it doesn't just wash off the slide. If you don't fix that organism and to the slide, it'll just rinse away during your staining process, and then you won't have anything to look at all. So in our lab, we're going to use heat. We're not going to really play with xylene or anything like that, although that is a great fixative, and I've used that in the real world. But in our micro student lab, we'll fix by heat, and so you'll learn to do that. So once you fix it by heat, then you can get into the actual staining process. So gram stain, again, is the stain that we pretty much use every day, day in and day out in the real world and in our student lab. So we use the gram stain to really differentiate our bacteria, to start to figure out what we have. Now, of course, we can't look under a microscope and be like, that's Staph aureus and that's E. coli. That we can't tell, but we can begin to figure out what group of bacteria might we be playing with, and then that will help guide our next steps. Everything in micro is kind of like a flow chart testing system. So once we know the gram stain, then we're like, okay, what's the next test under this side of the flow chart? And then we can keep moving. So we had learned last week briefly, gram stain really divides gram bacteria into either gram positives or gram negatives. And it's all dependent, again, on their cell wall makeup. Gram positives have a thick layer of peptidoglycan, along with teichoic acid cross-links that really embeds this purple dye. That's why gram-positives are purple. Gram-negatives, again, have a thinner layer of peptidoglycan. And so we'll see here as we start walking through the gram stain process how they become red, whereas the gram-positives remain purple. So those are the two big groups that the gram stain divides bacteria into, is gram-positives and gram-negatives. So again, this is what it's just saying, what I kind of discussed. Oh, I think, did I miss a slide? Yes, sorry, I missed a slide there. All right, so here's the overall steps. Again, we fixed the bacteria to the slide. In our lab, it's going to be through heat, but these are the actual 4 steps involved in the gram stain. And I have a spelling error there. There should only be 1 F there. It's crystal violet, then iodine. Decolorizer is your 3rd step, and then safranin is your 4th step. So those are the 4 reagents that we're using in gram stain. You absolutely need to know those. Crystal violet is our primary stain. As the name tells you, violet, it is a purple color reagent. Iodine is what we call our mordant, and that means it's going to bind that crystal violet dye to the bacterial cell wall. So after you've flooded your slide with crystal violet, and you'll do this in the student lab, so it'll make more sense when you actually get to do it. When you flood your slide with crystal violet, you let that sit for a minute, then you flood it with iodine, the mordant, that's going to really help bind that crystal violet dye into the cell wall. And you leave that sit for like a minute. And then you have your decolorizer. And this step is really crucial. This is where you'll first distinguish gram-positive bacteria from gram-negative bacteria. So because that gram-positive bacteria have the thick layer of peptidoglycan and teichoic acids, it will keep that crystal violet. Even after the decolorizer, it won't rinse it away. That crystal violet's really embedded into that thick layer of peptidoglycan, so it hangs on to it. Whereas gram-negative, because it had such a thin layer of peptidoglycan, once you do the decolorizer, it easily rinses away that crystal violet. It just clears it out of there because nothing was really keeping it hanging on. So now at that point, gram-negatives don't really have a color yet. They're just sitting there because you decolorized them. They were able to rinse it away. So they're sitting there kind of naked. Then your 4th step, the safranin comes in, and the safranin is a red-pink color. And so the gram-positive is already purple. It's going to maintain that dark purple color. So the safranin is not going to do anything to it. Whereas the gram-negative, like I said, is kind of naked at this point. The decolorizer washed away any of the other dyes. So it's open to be dyed. So the safranin will come along and stain that gram-negative bacteria. So this red color. And so that's why gram-negatives look red and gram-positives look purple. So that's kind of the overall process. You'll get to do this in lab, so it'll really help. You'll learn the timing of all of it. The decolorizer step takes a minute to get it right because it's just a few seconds that decolorizer is on there. It's easy to over decolorize or under decolorize. You will learn that in lab. It's very easy to go one way or the other. So it's kind of like fine tuning how long you really leave it. You'll learn all of that in the student lab. But again, you should definitely know your 4 reagents, and you should definitely remember the decolorizer is actually the step that will first distinguish gram-positives from gram-negatives and why that is with the cell wall. So again, this next slide talks about that cell wall. Again, as we learned last week, gram-positive cell walls, thick peptidoglycan with lots of teichoic acid cross-links. Again, that should be an I there. I love when I find my spelling errors. So teichoic acid cross-links are thought to be why, again, they really can embed that crystal violet color in there and resist decolorization. Gram-negative cell walls, a thinner layer. And then there are sometimes, depending on the sample you're gram staining, not everything is just pure organism culture only. A lot of times we might get in a sputum sample, a different swabs in from like a wound or something like that. There might be blood cultures we're gram staining. So there sometimes are cells that are also stained in the gram stain process. It's not just the bacteria that's sitting there. So if there are cells that are getting gram stained, like red cells, white blood cells, like neutrophils or something, those are all going to be red on the gram stain. So those cells will just all be red on the gram stain. They are not going to keep any of that crystal violet because they don't have like a peptidoglycan layer to embed it. So they'll just stay with the last stain possible, that last reagent possible, safranin. They'll keep that color. So any cells that are there, squamous epithelial or another one that you will see, they'll all be red in color, but you can still kind of sense what they are based on like how they appear, their overall look to them. If you've been through hematology, you already learned how to identify neutrophils. You kind of have an idea of what an epithelial cell looks like. I think I have a picture of one here to help us if you don't. But you might have seen one in your analysis, just not stained. So you kind of already have an idea of what they kind of look like. So you kind of get a gist of what they are when you see them, even though they're all like a pink color and they don't have the nice right stain for hematology, you'll still kind of know what they look like. So again, this is just a nice example of that difference between gram positives versus gram negatives, as we've been discussing. The thick layer, the teichoic acids here in the gram positive versus over here in the gram negative. They just have like a thin layer of peptidoglycan, like their peptidoglycan is right here. Again, the other main difference is gram negatives also have that outer membrane. Both positive and negative bacteria have cell membranes, but the gram negative bacteria also has an outer membrane that's right above that peptidoglycan layer, and gram positives don't have that. So that's another key difference we learned last week, just to keep in mind. So there's kind of the overall picture representation of gram staining and why one remains purple and the other remains red. Okay, so when you look at it under a microscope, you might have already seen some gram stain slides in week 1. I'm trying to remember. I think you might have. You'll definitely be staining every week, so you'll get used to seeing this. Here's on the left, that purple color. That's what we would call gram positive when I see this. And then here on the right is definitely that pink or red color. That's what we call gram negative. Again, we're not just reporting if it's positive or negative, we're also reporting shape. That's part of the morphology of doing a gram stain. So we will get into that here in a right now, actually. So under that 100x, you're looking for first the gram stain reaction. Like I said, you look at it and you're like, oh, it's positive or it's negative. And then you're looking at the morphology. Is it coxy, meaning round? Is it bacilli, which are rods? Or is it a spirochete, which is going to be kind of like a squiggle? We're not going to see spirochetes hardly ever. So just so you know, it's usually a coxy or bacilli. Or sometimes there's the fun coccobacilli, which it's ovally and it's kind of a mixture of the coxy and bacilli, because why would we ever have things simple in just 2 categories like that, you know? And then again, you can have special characteristics like, oh, they always are chaining, they're in clusters, they're in pairs. Sometimes you might report that. As we learn the different organisms, you'll start to hear about those special characteristics they might have. If you're looking at certain samples like a sputum, we definitely want to note if there's neutrophils present, because that again indicates there's an infection happening, a bacterial infection. Neutrophils' job is to eat bacteria, is to phagocytize bacteria, eat them up and kill them off. That's the main role of neutrophils, is to kill bacteria. So if you see an elevated number of neutrophils, that's usually a sign of infection. And so that's something, especially when we look at sputum samples, and we'll learn this again more micro too, we count and look to see if there's neutrophils present. That's part of that process. And then squamous epithelial cells, that epithelial is skin. Squamous epithelial cells make up a lot of our skin, our mucous membrane surfaces. We will look for these, especially again in respiratory samples like sputum, because they tell us, oh, it's contaminated. It's not a true deep lung sample. So some of you might be like, what is sputum? Sputum is that nasty loogie from deep in the lungs. If you think of somebody that's had bronchitis, pneumonia, they cough up some good stuff, like some mucusy stuff that I hate the sound of it. It's probably my worst sound I can think of to hear. I don't like getting it in the lab. I would rather play with stool than sputum. Sputum kind of grosses me out. That is one thing I like, because it's that loogie, mucusy, green. It's nasty. That's sputum. It's not spit. We all have saliva in our mouth. We all have spit. Sometimes we're like, oh, I need to spit. You know the difference between spit from your mouth versus you are sick and you are coughing up that deep lung sample. That is sputum. So the difference is squamous epithelial is going to be found in saliva and spit, because it's just coming off of the insides of your cheeks. And so you'll see a lot of epithelial cells from the inside of your cheeks in saliva, versus when you get a lung sputum sample, that's not, you shouldn't find hardly anything for epithelial cells. There should not be any epithelial cells. So that's a screen we do. And again, we will revisit this in micro 2. that's one thing why we look for epithelial cells. Epithelial cells tell us that's a contaminated sample and that's just spit. It's not your true actual sample that you want. So that's an indicator why we might look for that. Okay, so again, here's the bacteria morphology. It's a nice 3D representation, little cocci, so little balls there. Bacilli, which again are rod shaped, and then you have your spirochete, which is, like I said, I just call it a squiggle. I just think of it as a squiggle. So here's another, this is more of a flat cartoony representation. So here's where I talk about, like, you might indicate special arrangements like clusters. So Staphylococci, which will be the first group of bacteria we learn in a couple weeks, they tend to cluster. We call them like grapes. If you think of grape-like clusters, because they're round, they're cocci, and they occur in clusters. Whereas Streptococci over here on the right tend to chain. And you can see the difference between chain versus cluster there in cocci. Diplococci, diplo means pairs. They like to be in pairs. So those are some of the special arrangements that you might note when you're reporting a gram stain. Again, bacilli are rods. You have a nice little coccobacilli, which are ovoid. They might look like the between of cocci and bacilli. And then you have your spirochetes down here. So they can kind of take a few different looks depending on which type of spirochete you're playing with. But again, when you report out a gram stain morphology on every single lab report, you must report out gram positive shape, gram negative shape. I don't want to just see gram positive because I'm going to say to you, gram positive what? Is it gram positive cocci or bacilli? There are two very different realms of bacteria. Is it gram negative cocci or bacilli? Again, very different bacteria that occur in each one. So you must report out both the positive or negative and then what shape you saw. And then if you did see it in pairs, you can say diplo, you know, or something like that. But typically always at least say cocci or bacilli. And so here's an example of how they can be grouped up. When I say to you, if you just tell me gram positive, I'm going to say cocci or bacilli because here's my main gram positive cocci, but here's a lot of my main gram positive bacilli. You can see these are very different organisms, and I'm going to be thinking of them differently when I look for it. So again, this is the gram stain is all to help you start figuring out what might I be playing with here. If you get gram positive cocci, your mind should be right away thinking I either have staph or strep or enterococci. Those are my main players. So I know how to test for those. If you get a gram negative cocci, you're going to be like, I know exactly what I need to test for because there's not very many that I'm worried about with those. Again, this is not an inclusive list. These are just examples of bacteria that fit into each category, but it tells you how to test for them after your gram stain. All right, here's a look at those squamous epithelial cells that you might find. In case you've never seen what they look like, you should have in urinalysis, but again, in urinalysis, you didn't stain them. They would have been just unstained. But anytime we do gram stain, all cells will appear pink. They'll retain that safranin pink red color. So they just look like pink red. But squamous epithelia are very large cells. They have a lot of cytoplasm, one singular round nucleus, like you see here. They kind of look like fried eggs, if you think of it that way, a little bit. So that's what a squamous epithelial cell would look like under a gram stain, just as a note. All right, so that is gram staining, the most important stain and the one that you'll play with all quarter long. Other stains that you should note are acid-fast stains. We're not going to play with acid-fast stain because we don't have the bacteria that for those for a reason. We don't mess with that bacteria too much. Plus, the bacteria that usually we use for acid-fast stains are slow growers. They will take days, weeks to grow sometimes. So, but acid-fast stain is used because we do have bacteria that instead of having peptidoglycan and all that good stuff in their cells walls, they have mycolic acids instead. They are made up differently in their cell walls, so they don't stain correctly with gram stain. Gram stain just doesn't work on them. So as a result, we can look at acid-fast staining. So the acid-fast stain will target those mycolic acids that are in the cell wall instead. So here's just a little picture down here of, mycolic acid in the cell wall instead of having peptidoglycan. The main bacteria that we use acid-fast stain for is called mycobacteria. That is a group of bacteria. Myco, if you think of it as in the name for mycolic acids, if you want to think of it that way. So these bacteria all have walls that have mycolic acids in them. The bacteria you probably would have heard of that belong in the mycobacteria group is tuberculosis. So you guys have all heard of the disease tuberculosis, I'm sure, at some point, whether it's through a movie or maybe you've known someone that's had it. So that is the main bacteria that occurs, or that you would be familiar with in mycobacteria. Otherwise, there's a bunch of others that occur through different environmental stuff. All right, so again, acid-fast stain to target mycolic acids. The reason it's called acid-fast is that these mycolic acids are able to resist an acid decolorizer. So the decolorizer that's used in the staining process is an acid decolorizer, and they can resist it. They don't get decolorized with it, so that's why they're acid-fast. That's that name. All right, so there's kind of two methods to acid-fast stain. We have first the Ziehl-Neelsen method, and then the second one is called the Kinyon method. Very similar. There's one major difference between the two. So you're going to first make your smear, you fix it with heat so that your bacteria don't slide off the smear when you go to stain it. That step's still the same, like in the beginning. You're still doing that whole fixing the smear. Then you have 3 steps only. So you have a carbofuchsin, which is the primary stain. Carbofuchsin is like a pink or red color. I always think of the fuchsin as fuchsia, and I know fuchsia is kind of like a brighter pink purple. That's what helps me remember that color. So the primary stain here is called carbofuchsin. Now in the Ziehl-Neelsen method, once you flood the slide with carbofuchsin, then you're going to heat it to boiling and let it sit for 5 minutes afterwards. And the idea is that it really just embeds that carbofuchsin into the cell wall. So that heating boiling process here, so this is a different thing than when you first fix your smear. We're boiling this. You're embedding that carbofuchsin down into that cell wall. Then you apply the acid decolorizer. And again, if it is a bacteria that has mycolic acids like a mycobacteria, it will keep the carbofuchsin. It will not let the acid decolorizer wash it away. And then you apply methylene blue, which is our counter stain here. So anything that was acid-fast positive will maintain this bright carbofuchsin purple pink color. Anything that's not acid-fast will rinse it away and will be a blue color at the end. So again, that's Ziehl-Neelsen. The main difference with Kinyon is it's called our cold method. We do not boil it. We remove out the boiling process in the carbofuchsin layer, and instead they have a stain that has a higher concentration of phenol, and that instead embeds the carbofuchsin into the cell wall by having that higher concentration of phenol. So same reagents, they both use carbofuchsin, decolorizer, methylene blue, both of them. Again, the difference is Ziehl-Neelsen boils it in the carbofuchsin, whereas Kinyon has a higher amount of phenol in that reagent, so it doesn't have to boil it. It's a cold method. So again, interpretation, any bacteria that are considered acid-fast that do have the mycolic acids are going to be red or bright pink, if you will. Non-acid-fast are going to be blue. So we kind of have that here, this representation here. Before staining, neither one were stained. After they apply the carbofuchsin layer, they both are like this pink purple color. Then they decolorize it. Again, acid-fast have really embedded that fuchsia color into the cell wall. So they are acid-fast. They don't respond to the decolorization, whereas in non-acid-fast, they will wash it away. And then when they apply the methylene blue, they stay fuchsia still, whereas the others will pick up that blue color. So if we look at a sample, a lot of times, like, so this is kind of like a tissue gram stain. You can see there's a lot of stuff here in the background. Most of this kept that blue color, but again, these little organisms here, you can see a rod here, one up here, one right there. There's one down here. You can see those are a pink color. That is the bacteria there. We would say that is acid-fast positive. All the rest of this is just part of the tissue sample that was used in this stain. So you can ignore that. But yeah, these like bright pink red rods here are saying that is an acid-fast bacteria in it. All right, so that is the other stain to know there. Some other microscopy. So again, both acid-fast gram stain, we're using the bright field microscope, which is our main one. You can use phase contrast microscopy in the microlab. Typically, these are not stained items. You are looking at live, living, samples that are not stained. The way that phase contrast works is beams of light will pass through the specimen, but they're kind of partially deflected by the stuff that's in the sample, by the cells that are in the sample or the fungi that's in the sample. Again, you do not stain any of the sample when you use phase contrast. A lot of times it's used with maybe fungi identification. So that's the main purpose there for phase contrast. We're not using it for bacteria, not at all. There's also fluorescent microscopy, and just like the name tells you, we're using fluorescent dyes. So when you put a fluorescent dye on something, it raises to a high energy level after it absorbs UV light, and that once it establishes that high energy level, it fluoresces as it comes back down. So then when you look at it under the microscope, you're going to see if it's positive, if it fluoresced, it will show that under that microscope. With the fluorescent stain, there are two different categories. We have fluorochrome stains, which just use fluorescent dyes, and then you have immunofluorescence, which means the fluorescent dyes are linked with an antibody. Anytime you see the word immuno whatever, it's always dealing with antibodies. Anytime they put the word immuno in a term, it's saying that there's antibodies involved. Just a note there. So here I have kind of a visual representation. I know it's a little cloudy or fuzzy. Fuzzy would be a better word. But again, fluorochromes, they're just the dye itself that are looking for the bacteria to stain. Once they see that, they attach on and they light up, they fluoresce. Whereas immunofluorescence is a dye attached to an antibody, and then it goes and looks for the antigen to the antibody. You will learn all about that in immunology. But once it finds its antigen of choice, then it attaches on and then it fluoresces. There are a few fluorochrome dyes I want you to know. There's 3 of them. The first one is acridine orange, and you might get the chance if you work in a larger microlab to play with this. So acridine orange looks for nucleic acids, DNA, if you will. A lot of times this might be used in labs to look for bacteria, to confirm the presence of bacteria in blood cultures. So if somebody comes in and they're really sick with, say, high fever, all the signs of infection, maybe they have a pneumonia, a lot of times we're worried about that, has that infection spread to their blood to cause sepsis. And you should never have an infection in your blood. Your blood should never have bacteria in it, ever. That is significant. That is serious when that happens, because your blood travels everywhere in your body. So you think about it, then you're spreading the infection all over your body. So we draw blood cultures. If you've been a phlebotomist, you know what I'm talking about. But if you haven't, you've never seen that. And we will again revisit blood cultures in Micro 2. And you probably have learned about blood cultures maybe in a phlebotomy class. But we draw these blood cultures screening for sepsis, screening for bacteria that might have gotten into the bloodstream. So then we put them in a nice incubator in the micro department, and the incubator will alarm if they sense there's bacteria in there. It will alarm. So then you pull it out and you have to stain it and look for the bacteria. Sometimes they are a bugger to find in a gram stain, and you can't find them, but you feel like they might be there. Some labs will use acridine orange because the acridine orange seeks out the DNA in the bacteria, and then it fluoresces that, yep, it's here. You have bacteria here. So that's one of the main reasons it's used. It also can be used for, we have bacteria that do not have cell walls. If they do not have a cell wall, you can't gram stain them because the gram stain stains cell walls. So that doesn't work. So again, but cell wall deficient bacteria have DNA. So again, you can use acridine orange to look for that DNA and fluoresce. So those are certain purposes on why we would use acridine orange. The name tells you what color is it going to fluoresce. It's going to fluoresce orange when it's found there. So you should know that acridine orange is used to bind to nucleic acid, to DNA and bacteria. That's what I would remember. Here's how it looks. How cute. Like, right? I don't know why, but when I see that, I just think it's cute. Nice bright orange. Like, that's obvious. Like, oh, I got bacteria. That is obvious. You don't have to search for it. Like, that is just, it jumps out at you when it fluoresces like that. The second fluorochrome dye I want you to know is auramine rhodamine. This one looks for mycolic acid. So same purpose as acid fast staining. We're looking for the mycolic acids in mycobacteria cell walls. So once it binds to the mycolic acids, it will fluoresce a bright yellow or orange. So again, it's used for the same purpose as acid fast. So on the left here is the Ziehl-Neelsen acid fast stain, where again, it's a red color when they're present. So there's the arrow there. There's a group up here. On the right is showing you the auramine rhodamine fluorescing. So they just put one random arrow there, but you get the idea. So yellow, orange, fluorescent when it finds and binds to mycolic acids. So know that for auramine, rhodamine. And then the third one is calcofluor white. This one is looking for the cell walls of fungi. So it's looking to detect fungi in samples. I always think of the word calcofluor as cauliflower. And for some reason, cauliflower makes me remember fungi in my brain. I love cauliflower. I don't think it's a fungus, but like it seems like a fungi thing. So that's how I link it in my brain. So again, as the name tells you, it's going to fluoresce a white when it binds into those cell walls. So here, this is a yeast. Yeast are also fungi. It's not just molds. Molds and yeast both. This is a yeast that is found in a sample and lit it up. It's supposed to be a white, but it kind of looks almost green here. I'm sorry. Hold on. My dogs are, of course, playing now. So that lights it up a kind of a nice white color when it finds it. All right. So, and then like I said, there's immunofluorescence that use an antibody that looks for different antigens. We can look for any bacteria that might be hard to grow in a real world lab. Like some bacteria just do not grow on agar plates like normal bacteria. They just are not going to. And you might use a dye instead. I'm not going to really test you on immunofluorescence at all. So do not worry about this slide. All right. Another type of microscopy is dark field microscopy. It's basically the condenser on this does not let light pass directly through the sample. So that's why it's dark field. Instead, it hits it at a kind of an angle. And so it only hits objects like any organisms that are in there. And then it deflects that light upwards. So the dark background is dark, very limited for normal bacteria. We never use this for typical bacteria. We might use it though for spirochetes. Spirochetes do not grow like normal bacteria. They are not played with like normal bacteria. That's why I said we're not gram staining and looking for spirochetes necessarily. If we think we have a spirochete, we would use a different technique. Probably the main spirochete you've heard of and you'll learn a lot more of in immunology is Treponema pallidum. That is syphilis. You guys have all heard of syphilis, so that is a spirochete. We test for syphilis in a much different way. We don't even necessarily use a dark field microscope. We would use first, there is a screening test that you would run, and then if that's positive, you could go to a dark field confirmatory test. Other spirochetes that you've probably heard of, Lyme disease. Lyme disease is caused by a spirochete. So that's usually an antibody kind of test. So they have different testing purposes out there, but one way we can is to detect spirochetes. So there they are, again, the little squiggle shape. You can see the dark background. All right, and then your last microscope is the electron microscope. This is used to visualize very, very tiny little objects into a magnification in excess of 100,000 times magnified. So very, very tiny little structures. Most microlabs do not own an electron microscope. We're talking major, major labs, research things like that, that you would ever get to play with this. There are two different types, transmission and scanning. One gives you internal structures. The other gives you like an outward 3D view. And so there you go. That's kind of internal structures versus an outward 3D view. If you ever want to look up like some fun pictures of you, look up electron microscope. Like they have ones that they look like these crazy alien worms like out of Dune or something. There's some things on there that you can find electron microscope pictures. There's some really fun pictures on there on Google if you ever look up and want to play with. All right, so that is our first PowerPoint on microscopic examination. Lots of stuff to remember about gram stain, acid fast stain, those 3 fluorochrome dyes that we discussed there as well are probably the main purpose. And then just knowing what each microscope does, kind of like the gist of each microscope in general. All right, let's switch over to the colony morphology. This is probably an even bigger amount of information to take in. This is where we're going to really discuss all the different agar plates. And we're not even discussing all of them. There are so many that exist. We're just doing a big roundup of some. So lots of information in this one, lots of test questions that come out of this PowerPoint in this material. So there is 3 purposes of why we cultivate bacteria. That's a fancy word for why do we grow bacteria on an agar plate? Well, the cultivation is to grow and isolate any bacteria that might be present in that sample. So if you get in a urine culture, a wound culture, an ear, eye culture, anything like that of a possible infection, we're going to grow it to look to see, is there a pathogen? Is there something here causing the problem? So when we grow and isolate, it also determines which of the bacteria that grow are most likely the pathogen causing the infection. and what's just a contamination, what's just, oh, that's from the skin or something. And then the other purpose is, of course, to give us enough of the bacteria so that we can play with it and identify it. So again, cultivation just means the process of growing the bacteria in culture. So by taking it from the infection site, you know, with whatever specimen they sent you, and then growing it out in the lab. So in order to grow it, we have to give it an agar or a media in general that it likes. We call it media because it can be an agar or broth, and it gives it nutrients so it can thrive and grow. There are 4 categories of media: enrichment, supportive, selective, differential. You need to know the definition of each of these categories of media. So supportive sounds the way it does. It supports the growth of pretty much almost anything. Like sheep blood agar is our #1 most commonly used agar in the lab, is sheep blood agar. You might see the initials SBA for sheep blood agar, or you might see the initials BPA, blood plate agar, or BAP, sorry, BAP, blood agar plate. They're the same thing. It's sheep blood agar. So blood agar plate, sheep blood agar, same thing. #1 used agar in the lab because it's supportive. It's a great plate to grow pretty much everything, except fastidious organisms. Now, I did have a conversation with a student, and I've had this question every time I teach, and it's a good question because in the textbook and on other resources, you will read that blood agar is also enriched. So whereas it could grow fastidious organisms, some fastidious organisms, and there's the key word, some. So what I have chosen to do to teach this is we're not going to consider sheep blood growing fastidious. We are just going to say it grows pretty much most bacteria except fastidious picky organisms. Remember, the word fastidious means that is a very picky organism. It wants extra nutrients to grow. It doesn't just want the sheep blood. It wants more. It needs more than that to grow. It doesn't, you know, so it's not just going to grow on only sheep blood. So I teach this for a reason, because when we get to learning about certain fastidious organisms, you will learn they do not grow on sheep blood because it doesn't give it enough of the nutrients. And so I don't want to just go with this whole, oh, sometimes it can grow some fastidious, but not others. You know, nothing is ever perfect, but the way we're going to learn this is sheep blood agar is only supportive. It grows pretty much everything except fastidious stuff. It won't grow those. Let's just keep it to that. It's easier as we learn this. So then enrichment media is like it sounds. It's enriched. It has extra nutrients in there to grow those particular bacteria. If you see the word particular, it's underlined there. It's again standing for picky, fastidious bacteria. The 2 most common fastidious bacteria are going to be Neisseria and Haemophilus. We will definitely learn about both of those later this quarter. And you will learn they do not grow on normal supportive agar. They want the enriched agar. So that's why we keep them categorized a little differently. So enriched media, extra nutrients to grow fastidious bacteria. Selective media is only going to selectively grow certain bacteria. So it's picking out a type of bacteria to grow and inhibiting all others. So in selective media and agar plates that are selective, they have antibiotics or certain chemicals that will ensure that certain types of bacteria do not grow, whereas others will grow. So an example of this is PEA agar. That phenylethyl alcohol that is added in there inhibits gram-neg rods from growing and will allow us to grow gram-positive bacteria. So that's why it's selective. It's selectively growing the gram-positive and it's inhibiting the gram-negatives. So it's giving us a certain type only. And then differential means that there is, again, different dyes, things that are inside of that agar that allow extra characteristics to be exhibited. So once you grow whatever bacteria on that agar, differential will tell you it has different characteristics that can be used to distinguish them from other organisms on the plate. So one example of this is MacConkey agar, in that the dye that's in MacConkey agar, for those that ferment lactose, will be purple, and those that do not ferment lactose are clear. And so we're going to see this again when we get to MacConkey slide, but that's meaning differential. It differentiates the bacteria that are growing from each other. So those bacteria that grow will have certain characteristics that, yep, these are lactose fermenters, these are non. It can help differentiate between them. And so that's differential. So it allows one bacterial species to exhibit certain culture characteristics to distinguish them from others growing on that agar plate. So those are the definitions of the 4 categories that you need to know. All right, now let's get into the initial agars that we're going to learn, and you'll have a whole assignment that you'll do for this as well. Again, sheep blood agar, otherwise known as blood agar plate, #1 agar that we use in the micro lab because, again, it's just a great supportive agar. It grows almost everything except Fastidious. So here's what it kind of contains in there to give it some nutrition that it likes. The other unique thing that sheep blood agar has the ability to do is show hemolysis patterns on the plate. And you will get to, when you start learning bacteria and looking for bacteria on agars, you'll learn how to always look for a hemolysis pattern on the sheep blood agar. So certain bacteria will produce enzymes that lyse the red blood cells, that 5% sheep blood that's in that agar, that will lyse those cells, that will, wipe them out. So if we call something beta hemolytic, a beta hemolysis, that means they had enzymes that completely cleared the red cells around the colony of that bacteria. It's completely clear around it because they lysed it all. Whereas there's some bacteria that do alpha hemolysis. They partially lyse the cells. They don't fully wipe them out around the colony. They partially do. And as a result, it gives it a green look to it. And then gamma hemolysis means nothing. They did not do this at all. The agar is left alone. There's no hemolysis. So a sheep blood agar is going to fit into the categories of supportive, because again, it supports the growth of almost everything, and differential. And the differential is because it can separate between the hemolytic patterns. Differential is all about separating, distinguishing based on culture characteristics. Well, bacteria can take on different hemolytic patterns, that is characteristics. So it can differentiate based on that hemolysis. So those are the 2 categories that you should always place sheep blood agar into. So let's take a look at those hemolysis. So up in the upper left is beta hemolytic. So each one of these colonies is bacteria. And as you can see, this bacteria had enzymes that completely lysed away that red blood agar around it. So that's why they're glowing. They kind of look halo-like. They glow. In order to read for hemolysis, you hold your plate up to the light and you kind of look for that clearing through it. So when you hold your agar up to the light, you can look for the clearing around the colonies to see if there's beta hemolysis. And we would report that. That's part of certain organisms always are beta hemolytic and certain organisms are not. And so that's part of determining what we have. So you would report beta hemolysis if you were to hold this up to the light and look for it and observe. This one's hard to see. It's not showing great in the lab. You'll see it at some point. I should have found a better picture here. But the one to the upper right here is supposed to be alpha hemolysis. Again, if you hold that plate up to the light and look through it, these around each bacteria should be green. They should have a greenish hue to them. They're not going to be nice and clear halo like this. They're going to have a green hue. That is alpha hemolysis. And then down here is a good representation of gamma. No hemolysis at all. You'd hold this up and you don't see anything happening. There is no green effect. There's no halo effect. It's just gamma, no hemolysis at all. All right. Brain heart infusion agar is an agar that is, again, a supportive agar. I don't care about it. I'm not going to talk about it. We're never going to play with it really. So it's just mentioned here. All right. Chocolate agar, very commonly used. Chocolate agar is supportive because it grows pretty much everything, just like blood agar, but it's also enriched. So it has extra nutrients. So therefore is great for fastidious bacteria. It is an excellent agar for those picky bacteria. So again, like I said, Neisseria and Haemophilus are 2 very well-known fastidious bacteria. They will not grow on sheep blood agar. They will grow on chocolate because it's giving them that enriched, plates. So that's a key thing there. So the reason that chocolate agar is enriched, it is the same makeup as blood agar. It also has blood in it, 5% sheep blood. But when they make it, they lyse the red cells on purpose, which makes it this chocolate look. It looks like chocolate. And when they lyse those red cells release out hemin, which we call an X factor, and then NAD, which we call the V factor. You will see these mentioned again when we learn about Haemophilus later this quarter. So they release out these extra nutrients, this X factor and this V factor, and that's what Neisseria and Haemophilus use to thrive. They're like, oh, we really like that. And then they'll grow. So chocolate, supportive, and enrichment. Thioglycolate broth is, like it says, a broth. It's a liquid, so it's got great nutrients in there. So it's both supportive and enrichment. It has extra nutrients as well, so to grow pretty much everything in that environment, in that broth. It's kind of neat though, because certain bacteria will take on different looks in the broth when they grow. Last week we mentioned that when you have bacteria present in broth, it will make it cloudy or turbid looking. So different bacteria will look different cloudy or turbid ways in the thio broth depending on what they are. If they are gram-negative bacteria, they will appear cloudy throughout the entire broth, or what we call diffuse. The entire broth will be cloudy or diffuse looking if it's more of a gram-negative organism. If it's a gram-positive cocci or balls, it kind of can sometimes take these cloudy little spheres or ball shape on in the broth. So we have a picture here. So here you can see letter B there is trying to show you those cloudy little, they look chunky, like, but if you think of it as like cloudy little balls, like that is telling you maybe that's a gram-positive cocci grind. It is not picture perfect by any means, but it's a good indicator. You would of course just realize, oh, something grew in here and you're going to play with it, but that's just showing it. Whereas C is showing you the cloudy, oh, I think I know what C is showing you, but anyway, so the cloudy there. The other thing to note, did I not? Hold on. Okay. The other thing to note is it can also grow anaerobic bacteria, whereas anaerobes do not want oxygen at all. If the anaerobes are like letter D here, so the broth is all the way up to the label there, but you see it's only cloudy at the very bottom. That is telling us we might have an anaerobe growing because it did not want to be at that surface at all. It wanted to be as far away from the surface as possible. So it only was cloudy at the bottom. So that's another indicator. So if you have turbid or cloudy growth at the bottom of the tube only, that's most likely an anaerobe. Whereas the opposite, if you have bacteria that are strict aerobes, meaning they have to have oxygen to grow, they're going to then be cloudy at the top only. They're going to be as close to that oxygen as possible to be growing. So strict aerobes, cloudy at the top only. Anaerobes, cloudy at the bottom. Gram-negatives, cloudy throughout, diffuse throughout. Gram-positive cocci, little cloudy balls, if you will. So those are kind of some unique features of thio broth. Again, it's not a picture perfect system. It doesn't work like that always, but when it does, it's great. But again, supportive and enrichment. It's set up to pretty much grow almost pretty much everything. Okay, next one is Columbia CNA. We just call it CNA. It has an antibiotics colistin and nalidixic acid in it. These are added to inhibit gram-negative bacteria. So it's only going to grow gram-positive bacteria. That is all it will grow is gram-positive. So if you ever see growth on a CNA plate, you're going to know it's a gram-positive for sure. You still have to gram stain it, of course, but it should be gram-positive. So as a result, it only is selective. Selective because it's selectively just growing that gram-positive. That's the only category it fits into. Same with PEA, same use. It has phenylethyl alcohol in it to inhibit gram-negatives, so it grows gram-positives. So it's selective. Labs will most likely just have one or the other. They're either going to have a CNA, that's what they want in, or PEA. Some labs might have both as options, but a lot of labs are like one type and just order that in because they're used for the same purpose to grow gram-positives. There's also another agar out there called rose agar. It's a combination, it has both CNA and PEA in it. Again, it's selective, is looking just for gram-positives. There's a ton of different agars out there. Like I said, we're just mentioning some of the main ones. All right, McConkey agar is selective because it is only growing gram-negative rods, gram-negative bacilli. So that's the selective reason. And it's also differential because it will tell us if we have lactose fermenters versus non-lactose fermenters. So it differentiates those characteristics of the colonies. So it has this crystal violet dye in it, and that dye, crystal violet, violet for like a purple. So if it's a lactose fermenter, it will be purple colonies. If it's a non-lactose fermenter, they will remain colorless or clear. So that's kind of one thing when you look at a MacConkey agar plate. One, you always know right away it's a gram-neg rod in there growing. And then you're looking at, do I have purple colonies or do I have clear colonies? You will want to report that out if you have a lactose fermenter or a non-lactose fermenter, because again, it helps us determine what type of bacteria we have. So that is why it's selective and differential. Selective for only growing gram-neg rods, differential for the fermentation characteristics. So here is what it would look like. Lactose fermenting colonies are a purple, non-lactose remain clear, as you can see there. They say pink, doesn't matter, same thing. All right, EMB stands for eosin methylene blue agar. Same purpose as MacConkey. It is only going to grow gram-neg bacteria. So it's selective for that reason. It especially likes the grouping Enterobacterales. When you learn gram-negative bacteria, Enterobacterales, which we will learn about those in Micro 2, that's your main players. When you hear Salmonella, Shigella, E. coli, all of those are in that family, Enterobacterales. Those are your main culprits of problems. That's what it's used to look for. So again, it's looking for gram-negatives, so why it's selective. It's differential because again, it can be based on lactose fermenters versus non-lactose fermenters. So again, a lot of labs might have one or the other. They might like always having MacConkey agar in hand. They might like having EMB agar in hand. They have the same purpose. One note here is when they are lactose fermenters on EMB, they will take on a green metallic sheen. And you see that here. E. coli is well known to be a lactose fermenter, and it will have that beautiful green metallic sheen on it when it grows. Look how pretty that is. Hey, it's not fun to have E. coli, but it's pretty. So very pretty looking there. That green metallic sheen tells us that is a lactose fermenter. So I would remember that especially that E. coli also does that. And then non-lactose fermenters are just colorless on there. All right. Next agar, Hectoin enteric agar, otherwise known as HE agar. This has different, again, salts and dyes in it, so it's going to grow gram-negative rods, so it's selective, but it's really looking for Salmonella and Shigella. Both Salmonella and Shigella are gram-neg rods. They are known problems of the GI tract, your gastrointestinal tract. Lots of diarrhea, food poisoning, things like that. So when we think we are screening somebody who has, like, say, diarrhea, they come in and they're like, oh my God, I've had diarrhea for 3 days straight. We will probably get in a stool culture. You might set up an HE plate looking for, oh, do they have a Salmonella or Shigella infection causing this? So HE agar is great to screen for Salmonella and Shigella. So both of which are gram-negative rods. So that's the selective part. Again, the differential part is looking for fermentation characteristics. Salmonella and Shigella do not ferment lactose, so they will be a blue-green color on HE agar when they grow. So that is bolded for a reason there. The other thing to note is Salmonella will also produce hydrogen sulfide, what we call H2S. That will make it a black precipitate. So in the center, the colonies on the plate, they might show this black center, and that's because Salmonella can produce an H2S to make it black when it grows. Shigella does not. Shigella does not produce that, so it won't do that. So when we look at it here on HE agar, Shigella over here will be blue-green colonies. Salmonella is also blue-green colonies with that black center there for the H2S it can produce. So, but both are blue-green colonies because they are both non-fermenters. Those bacteria that do ferment will be orange on HE agar. So that's selective and differential. So know that, know the purpose of HE agar and the color that they take on. All right, the other one that's very similar to HE is XLD agar, known as xylose lysine desoxycholate. That's not an easy word to say. XLD, easier to say. Same purpose, looking for Shigella and Salmonella. So again, it's selective because it's looking for gram-negative rods, specifically Shigella and Salmonella. So again, most labs are going to either have an HE plate on hand or an XLD plate on hand. And then the differential part, same thing here, fermentation. Shigella and Salmonella will be colorless when they grow on here. And again, Salmonella might have that black center due to H2S production. Other things that grow are going to be different colors. Some sources might say that Salmonella can be a different color on XLD. let's just keep it in mind that they're colorless. Let's keep it simple for our world that they will be colorless colonies. But they could have that black center for H2S with Salmonella. So here is XLD agar. So pretty much colorless colonies. They don't have any color here to them. Again, a black precipitate in the center. So this looks like it's black, but it's colorless around the black precipitate for Salmonella. All right, so that is XLD. Next one is gram-negative broth. So this is another broth. The name tells you it's looking for gram-negatives, so it's for sure selective. That's it. I don't really care that much about gram-negative broth, so I don't say much about it. All right, the next two are Thayer Martin and Martin Lewis. Both are the same purpose. They are both looking for Neisseria. The main bad boys of Neisseria are Neisseria gonorrhea, which causes gonorrhea, and Neisseria meningitidis, which causes meningitis. There's a few things that cause meningitis, but Neisseria meningitidis is definitely one. So they are two major pathogens. Those are the two big pathogens of the Neisserias. We'll relearn that when we get to Neisserias. But Thayer Martin agar and Martin Lewis are used to specifically look for Neisseria. So that is why it's selective, is they're selectively looking for Neisseria. It is enriched because, again, Neisseria are fastidious. They are picky buggers. They want extra stuff. So they are enriched with extra nutrients to ensure the Neisseria will grow. Now, the way that they ensure that only Neisseria grows and nothing else is they have a bunch of antibiotics in the agar. So it's basically a chocolate agar filled with antibiotics. The chocolate agar gives them the extra nutrients to be enriched. They add in colistin to inhibit any other gram-negatives from growing, so that way no gram-neg rods or anything like that grow. They add in vancomycin, which inhibit gram-positive bacteria from growing. They add in nystatin into Thayer Martin to inhibit yeast from growing, and then they add in trimethoprim to really inhibit Proteus. You will learn more about Proteus in Micro 2. Proteus is just a gram-negative that really swarms and can take over a plate, and we don't want that. So that will help inhibit more of that Proteus from growing. So 4 different antibiotics added into the plate to ensure that only the Neisseria comes through and grows. So Thayer Martin, you need to know the purpose of it, what it's growing, the categories it's in, and the 4 antibiotics it has and what they inhibit. Now, Martin Lewis, same thing, looking for Neisseria. It's selective and enriched. It's just got a couple tweaks to the antibiotics. So instead of nystatin, it uses mycin to inhibit yeast. So Martin Lewis will have mycin to inhibit the yeast, not nystatin. And then it will have a higher amount of vancomycin. It upped the dosage of vancomycin in the agar. But again, both are the same purpose. Both are the same categories. There is a third agar that grows Neisseria called New York City. I call it New York City, NYC agar. And I'll re-mention this when we get to the chapter on Neisseria. But the way I always remember what these grow, not a great saying. Do with it what you will. But I always say, oh, the 2 Martins went to New York City and got gonorrhea. That's not a great saying. But it remembers that the Thayer Martin and Martin Lewis, the 2 Martins, are used to look for Neisseria. I always remember Neisseria gonorrhea. We also have meningitis. It's just an easier way to say it. But that's one way you can remember, because as you can tell, these are a lot of agars and it's a lot of memorization. There's just no way around it. It's just memorizing all of this, the categories, the purpose, what they have in them, that kind of thing. So that is one way that I help remember the Thayer Martin and Martin Lewis go to New York City and get gonorrhea. So I remember that they're used to grow Neisseria. And then I remember Neisseria is picky, so it has to be enriched. And then it's only growing Neisseria, so it has to be selective. Unfortunately, this is just memorization for the antibiotics in each. Okay, so that's the agars till now. Good news. We're going to learn more as we learn other organisms. You'll learn of other particular agars at that point, but that's the main gist of them for now that you have to know going forward. All right, so isolation of bacteria from samples. So to enhance the growth, isolation selection, we streak the sample across the agar plate. So again, you will do this in lab. It's really fun to learn how to streak agar plates. I think it's one of the most fun labs to play with and learn how to do this. So the goal of streaking across in a pattern is to get isolated individual colonies to look at. Otherwise, they're all going to be on top of each other. And when you go to test, you don't know if you're testing isolated colonies that are the same. You might be testing several different types of bacteria at the same time and not realize it. You need to have isolated colonies so that way when we test them, we know we're testing what we want to test. So we have a whole flame loop for sterilization and streaking quadrant technique. You are going to learn this in lab and do this. So ideally, we streak almost every culture into 4 quadrants. We have what we call primary, secondary, third, fourth, tertiary, quaternary. You get the idea. So your first, if you have a swab that has been sent down to you, say a wound specimen, a wound swab, you take and you make your primary quadrant with that wound. You just, you swab out the first primary, like right here. You see, it doesn't take up the whole plate. It's just a portion of the plate right here. Then you have what we call a loop that we sterilize into a Bacti-cinerator. So it heats it up, make sure the loop is sterilized. Once it's cooled off, then you go and you can streak out your secondary area. You got to make sure the loop is cool or you'll kill your bacteria. You will sizzle them out. You'll hear the sizzle if you don't make sure it's cool. So as you can see, what it does, and you'll learn this again in lab, is that you just go into the primary, pull out a couple of those strands, and then continue to streak. Then again, you're going to put the loop back into the Bacti-cinerator, let it sterilize it, and then you can go and make your third. Now, in this example, it's only made 3. There is a 4th one that gets made as well, and you'll again learn it in lab. If you look here, I know it's hard to see right now, but you'll see it on your own PowerPoint, is in the primary, you see how it's just, they're all mushed together. You can't tell those colonies apart. As they go, you can start to see isolated colonies. That is ideal. Out here, when we test, this is where we want to test, is right here in these isolated colony areas. All right, that is most culture plates are streaked to 4 quads. Urine cultures are the unique one out. They have a calibrated urine loop that we use instead. We streak once down, so you dip the calibrated loop into the urine, you streak once down the center, and then back and forth as many times as possible. The reason that we use a calibrated loop is we are counting the number of colonies present then. Because it's calibrated, we can figure out a number that goes with it. What we call colony forming units, CFUs. So in this case, letter B here shows you it grown out. This is a urine culture grown out. That's a lot of colonies it's growing. It's a McConkey plate here. I can tell by the look of it. But you might come in and be like, okay, I see. You count the colonies up. Well, in this case, you can tell there's over 100 because there's so many in these little built up lines here. So we would just say greater than 100,000 or something of lactose fermenting, whatever, you describe it. So we will learn that here. So basically, again, if it's just the standard streaking of 4 quads, when you look at it to report it out, like how much has grown, if it only grew in the primary, it's a 1+. If it grew to the secondary quad, it's a 2, to the third, 3+, to the fourth, 4+. And then you describe the colonies. Oh, I see 3+ medium grays. That you describe what you're seeing. And again, some agar plates are going to have more than 1 colony type growing. They're not just going to be pure always. So you have to figure out what's important and like, oh, I have 2+ beta hemolytic white colonies. You're describing what you see. You describe the size, the color, if it has a hemolysis, all of that goes into a description of what you see on that agar plate. And again, the 1+, 2+, 3+, 4+ stands for how much of it grew. Did it grow only in the primary, secondary, third, or fourth? The urines, again, are in number. Because you're using a calibrated loop, you can actually give a number here with it. So this is just an example. I have 50,000 CFU, coliform units per milliliter, because it's, again, calibrated, of medium grays, something like that. All right, so again, when you think about colony characteristics, size, color, shape, even if it has hemolysis, if it pits the agar, there's only a few that do that. Odor, do not put your nose to the agar plate. Back then we used to. You can do so a little bit because there's going to be a couple we'll talk about that smell kind of fun, but there's a lot that do not smell fun. Don't be putting your nose to the agar plate. Please don't, for the safety of all of us. We don't want to get up that close. It used to be that you can note an odor. Odor is very subjective. What I think something smells like, you're going to think smells different. So odor is not really a very objective description. It's subjective. Each person thinks it's different. So it can be noted, but it's not necessary. And then, of course, once we read the agar plate, we're like, we describe the colony morphology, then we gram stain it. And so you gram stain it just like we discussed, and then you report out your gram stain. Gram positive bacilli, gram negative bacilli, you know, something like that. All right, so again, all of this is going to go to helping us identify. So first you determine, do we have pathogen or contamination present? And that is based on what specimen type you got in. You will learn that as we go. It's not something we're going to learn right this minute. It will also help determine type of antibiotics to use. As you saw with the agar plates, we can provide vancomycin to treat gram-positive organisms. And the doctors know that when we start reporting out, micro is not a fast process all the time. So when we give them little snippets like, oh, we have gram-positive cocci growing, further identification to follow, they can already start using antibiotics. They might use a broad spectrum. We'll talk antibiotics next week, but they could also tailor it like, oh, I now know I have a gram-positive. I can use an antibiotic that's more tailored down to that. It just helps guide care for that patient. So overall, the identification process is the colony morphology on the plate, then the staining characteristics of the gram stain. Then we get into the biochemical testing, which we will start to learn as we learn about our different bacteria. And that will identify our bacteria, so identification then. And then we do what we call sensitivity testing, which is your antibiotic testing. And that we're going to learn more about next week. All right, that was, oh my gosh, a ton of information to know. This is a big chapter and lots of test questions come out of this PowerPoint. All right, thankfully, our third and final PowerPoint for the week is much shorter, briefer, thank goodness. So this one is on specimen collection and processing. I'm going to skip the objective slide there. You can read it. But your lab results, I'm sure you maybe have heard this in some other class, are only as good as the sample you get. If it was a poorly collected sample, you're not going to have great results. You might have contamination, especially in the micro world. So you will have provided to the nursing staff, to the providers, very specific collection directions. Depends on the site, the source. Is it a wound? Is it an eye? Is it an ear? Is it cerebrospinal fluid? Is it urine? Is it a biopsy, a tissue sample? All are very different. They all have different collection directions and what they should be collected on. We have different types of swabs. Certain things should be collected on this type of swab, others should not. We have, we have different containers that samples get put into. So all of that goes into the directions. We also want to ideally collect during the acute phase of the illness, meaning when they're actually really sick. They're actively sick before they got antibiotics. Once you start giving them antibiotics, the antibiotics are going to start trying to wipe out the bacteria. Then if you do a collection, we might not have anything grow because they've already started working. So we might not ever know what it was. So you ideally want to get your specimen before they give them antibiotics while they're actively at their sickest, because that is what is going to show up the most. So the micro department will provide, again, all the instructions to the provider's nursing staff. We are ultimately responsible for getting good, properly collected samples down. So again, this helps them with the selection of the appropriate site, the type of container or swab we want, how long it takes to get it down there. You can't just let stuff sit around on countertops forever. Some things maybe need to get refrigerated, like a urine. You can't let that sit out. It has to be refrigerated if we're not going to set it up right away. And then labeling, of course. So for stool samples, we would use a sterile container. It a lot of times has a preservative in the container to help preserve the stool sample. Swabs are going to be ideal for anything like ears, eyes, genital tract, upper respiratory tract, so like your throat, your nose, things like that. There are different swab types. Like I said, we are not using a typical cotton Q-tip. Cotton actually will kill certain organisms because of the fatty acids in it. So we might use a Dacron swab or calcium alginate swab. Urine, again, is going to go into a urine cup. Ideally, they're doing a clean catch midstream, so really good directions to the patients. You probably learned about this in your analysis. First morning is great because that's super concentrated, but it doesn't have to be first morning. Sputum, again, and that's a deep lung loogie. Definitely can be difficult to collect. Ideally, first morning is really preferred on sputum because that's a lot of times when they're sick, that's when they're coughing up that stuff is first in the morning. We do not want saliva because saliva will have a bunch of normal flora, bacteria from your mouth in it. That's just contamination. You could also do a bronchoalveolar lavage. That is a bronchoscope that gets inserted into lungs and then they collect the fluid. That could also work for assessing the lower respiratory tract. Again, this is usually looking for like pneumonia, what's causing pneumonia or something. Stool, you could do a rectal swab, but otherwise it's usually getting the stool sample into a container with preservatives. Also parasites. A lot of times people who have a lot of diarrhea, we're screening for both certain bacteria that are causing the diarrhea, but we're also screening for parasitic infection. So that's what we call an O&P exam, ova and parasite, ova for the eggs of the parasite. We learn all about parasites in Micro 2. But again, a lot of times when somebody has diarrhea, we give them a container for the stool for both just stool culture and for the parasite exam. So again, those containers have preservatives in there. This is all just information at this point, by the way. Again, transport, can't let stuff just sit around. Usually we want things within 2 hours of collection coming to us. They must be in a leak, please God, be in a leak-proof container in a plastic bag. There is special preservatives that can be given, especially if we think that it needs to be sitting for a minute. Chemicals like boric acid can be used to preserve urine. If we cannot get urine into the fridge and it needs to sit on a counter, we can use a preservative there to help it. And then there's other media here that will help maintain the viability of the organism if we can't get it set up right away. This, again, very informational. I do not test you on this. There's also anticoagulants. So if we're collecting blood samples, any fluid samples, we don't want them clotting and clumping. So there is anticoagulants in those specimens to make sure that it stays liquid so that we can play with it. We can't play with a clotted sample. It has to stay liquid. So again, this is informational. All right, ideally we refrigerate these items. These items could stay room temp just because of if they have preservatives or just the nature of what they are. Urine definitely has to get refrigerated if it's not preserved, if it's just in a normal urine cup. If it is preserved, then it could be room temp up to 24 hours. And then labeling. The biggest thing with labeling is if you learned phlebotomy, you learned about the importance of labeling, identification, right? Normally you have always the patient name, the patient's date of birth, the time, the date collected. But for micro, you have to have the source. If you send me a swab and do not tell me what it's on, it means little to me because your ear pathogens, the bacteria that are known to cause like ear infections versus what might cause a wound infection versus a genital tract swab. Genital tract swab is gonna be far different than an ear swab. And I need to know that because it helps determine what am I looking for? What could be the culprit? I know the bacteria that are going to cause genital tract infections versus the bacteria that are going to cause eye or ear infections. That helps us. We have to have that source. It's essential. It will get rejected if not. So that's all on the label. And then ideally, if it's like a wound, it's left foot, right ankle. I mean, that all goes to the care of the patient anyway. And then the requisition, which again, this is in the computer. The source again, the nature of the specimen, the diagnosis, if they've had any sort of immunizations or antibiotics given to them, that all can go into play. All right, specimen receiving. Once you receive in the lab, you mark it when you've received it. You prioritize it as necessary. We do a gram stain sometimes if we need to right away. Sputum, we always gram stain it right away. Other things we don't gram stain. We set it up onto plates and let it grow and then play with it. Just depends. And then there's, of course, always reasons why we reject stuff and say, we're not taking that. And these are all things. Again, this entire PowerPoint is pretty much informational. I'm not going to, as we learn about bacteria and different sites through these 2 quarters of micro, we learn more and more about this stuff. And then once you go to clinicals, you're going to live and breathe it. So this was just a nice informational one. We have plenty of stuff to remember from the first 2 PowerPoints. All right, so that is it for this week. Reach out if you have any questions.
Thank you.